Finding neurons that produce particular neurochemicals. It is of three ways. First one is localizing chemicals themselves. Second is localizing enzymes that produce them. Third is localizing the messenger RNA involved in this synthesis. Fourth is in situ hybridization, the production of DNA complementary to particular messenger RNA in order to detect the presence of RNA. Identifying neurons that produce specific neurochemicals is crucial for understanding the neural basis of behavior and cognition. Various techniques have been developed to achieve this goal. Immunohistochemistry is the first one, in which the first one is antibodies are proteins that bind to specific molecules, including neurotransmitters. By labeling brain tissue with antibodies against the particular neurotransmitters, researchers can visualize the neurons that produce or contain that neurotransmitter. Second thing is fluorescent dyes. The fluorescent dyes can be conjugated with antibodies to make them visible under microscope. This allows for precise localization of neurotransmitter producing neurons. Second is in situ hybridization. Proofs. This technique involves using proofs that bind to specific mRNA sequences. Since mRNA is precursor to proteins, including neurotransmitters, identifying neurons that express mRNA for a particular neurotransmitter indicates that they produce that neurotransmitter. Radio labeled probes. Radioactive isotopes can be used to label probes, allowing for detection using autoradiography. The fluorescent probes. The fluorescent dyes can be and also be used to label probes, allowing for visualization under microscope. Neurochemical assess. Microdialysis. This technique can be used to collect samples of extracellular fluid from specific brain regions. The collected samples can then be analyzed using techniques such as high performance liquid chromatography, HPLC, or mass spectrometry to quantify the levels of various neurotransmitters. In vivo voltammetry, this technique can be used to measure the release of neurotransmitters in real time. By implanting an electrode into a specific brain region and applying a voltage, researchers can detect the oxidation or reduction of neurotransmitters, which is proportional to their concentration. Optogenetics. Genetically re-engineered neurons. This technique involves genetically engineering neurons to express light sensitive proteins. By activating or inhibiting these neurons with light, researchers can identify and study the effects of their activity on the release of neurotransmitters. The challenges and limitations. The specificity ensuring that the techniques used are specific to target neurotransmitter and not affected by other substances can be challenging. Sensitivity. Some techniques may have limitations in terms of sensitivity, making it difficult to detect how levels of neurotransmitters. Dynamics. Neurotransmitter levels can fluctuate rapidly in response to various factors, making it difficult to capture their dynamics. Despite these challenges, the development of new techniques and advancements in analytical methods are enabling researchers to make significant progress in identifying neurons that produce particular neurochemicals. These methods are essential for understanding the underlying mechanisms of brain function and for developing treatments for neurological and psychiatric disorders. So, localizing particular receptors. Two different procedures are used in localizing the particular receptors. Two different procedures are radio auto autoradiography using a radioactive ligand that binds to receptor. Immunohistochemistry use protein antibodies to label the receptors. Through these methods, experimenter can not only determine the chemicals that a neuron produces, but also what connections that neuron has with other parts of the brain. This is called double labeling.